Hello, everyone. Have you ever stopped to think about what's running your body in the background? On autopilot? There is a very powerful, very complex system that's working all the time. A real multitasker that you don't even have to think about. So today, we're going to demystify that system, the autonomic nervous system.
Just think about it for a second. Right before a big exam, or maybe an interview, your heart starts pounding, your palms get all sweaty, or you're just walking past a canteen and you smell some hot samosas frying, and immediately your mouth starts watering. Now, you didn't consciously tell your body to do any of that, right? That is the autonomic nervous system, or the ANS at work. It's your body's amazing autopilot. So here's our roadmap for today. We'll start with the big picture, this autopilot system. Then we'll meet its two main teams, one for getting you ready for action and one for chilling out. After that, we'll figure out how these nerves actually talk to each other. We'll put a spotlight on a real star chemical messenger. And finally, we'll see why it's never a one-man show.
Okay, first things first, let's get our bearings. To really understand the autonomic system, we need to see exactly where it fits into the grand scheme of your body's entire nervous system.
You see, our nervous system has two major parts. There's the somatic system, which is what you control consciously, like deciding to raise your hand to ask a question. But the other part is the autonomic system. This is the involuntary one, the autopilot, controlling your heart, your blood vessels, your glands, all without you even thinking about it. You don't have to remind your body to digest your lunch, do you? It just happens.
So, this autopilot system isn't just one single thing. It's actually split into two divisions. And you can think of them like two competing teams inside your body, both absolutely essential for keeping you alive.
And these two teams have some pretty important differences. The sympathetic division, that's your fight or flight system for emergencies and stress. Its nerves come from the middle part of your spinal cord, and its main chemical messenger is noradrenaline. On the other hand, you have the parasympathetic division. This is your rest and digest mode for conserving energy. Its nerves come from the brain and the very bottom of the spine, and its superstar messenger is acetylcholine. Really, you can just think of them as the accelerator and the brake for your body.
Okay, so these nerves are controlling everything. But how? How do they actually send their messages? How does a nerve tell your heart to beat faster or your stomach to digest? It's not magic, Yar. It's all chemistry. Let's uncover their secret language.
The key concept, the funda, is something called neurohumoral transmission. Now, it sounds very complicated, but the idea is actually very simple. Nerves don't physically touch the thing they're talking to. Instead, they stop at a tiny little gap, which we call a synapse, and they release a chemical messenger to carry the signal across that gap. It's kind of like passing a note in class.
And this whole process can be broken down into four very logical steps. First, you have an electrical signal, an impulse, that zips down the nerve. When it reaches the end, step 2, it triggers the release of the chemical transmitter. Then, step 3, this transmitter floats across the gap and acts on the next cell. And finally, step 4, to get ready for the next signal, the message has to be stopped, it has to be cleared away.
So, what happens when that messenger hits the target cell? Well, there are two basic outcomes. The message can be excitatory, what we call an EPSP, which basically tells the next cell, go, fire, do something. Or the message can be inhibitory, an IPSP, which tells the cell, stop, don't fire. Simple as that. And that final step, how does the signal actually stop? I mean, the message has to be cleared away, otherwise the cell would just keep firing forever, right? There are a few ways this happens. Sometimes the messenger is just destroyed by an enzyme. Other times, like with noradrenaline, it's taken back up into the nerve to be recycled and used again. And sometimes, it just simply drifts away.
All right, now let's put the spotlight on one of the most important chemical messengers in your entire body. The absolute star of the parasympathetic system, acetylcholine, or as we call it, ACH. So how is this star messenger actually made? It happens right there, inside the nerve ending. The cell has to pull in a substance called choline from the outside. And listen carefully, this is the most important rate-limiting step. It's the bottleneck. No choline means no ACH. It's that simple. Once inside, an enzyme combines it with another molecule, and the final product, ACH, is packed into tiny little bubbles called vesicles, all ready for action. Now, what's really crucial here is that this very precise release mechanism can be a target. For example, botulinum toxin. Yes, the same one they use in Botox. It physically blocks the release of ACH. No release means no signal, and that causes muscle paralysis. And on the complete other extreme, you have the venom from a black widow spider. It does the opposite. It causes a massive, uncontrolled release of all the ACH, which completely drains the stores and leads to a whole different kind of problem.
Now, for a very long time, the simple model was one nerve, one chemical messenger. We thought a cholinergic nerve only releases acetylcholine. An adrenergic nerve only releases noradrenaline. But as we've learned, the story is actually much more interesting, much more complex than that.
And this brings us to the concept of code transmission. Basically, it means that when a nerve fires, it doesn't just release its main primary transmitter like ACH or noradrenaline, it also releases other substances at the same time, things like ATP or various peptides. It's like a chemical cocktail. And here's how it works. The nerve terminal holds the primary transmitter in some bubbles and the co-transmitter in others. When the nerve fires, both are released. Then they go and act on different separate receptors on the target cell. This allows the co-transmitter to modify or fine-tune the main signal, making the whole system much more sophisticated.
I'm telling you, this discovery was a real game changer. These co-transmitters can make the main signal stronger or weaker. They can act as a completely alternate message, and they often work on a different time scale. Some are super fast, others are slow and long-lasting. This revelation of co-transmission has helped explain so many things in pharmacology that were confusing us for years.
And all of this brings us to a final and very powerful question. Now that we understand this incredibly intricate system of chemical messages, what happens if we can design drugs to block them, or mimic them, or change how they're released? Just think about the possibilities in medicine. And that idea, that is the very foundation of pharmacology, which is a topic for our next session.
Namaste Bachcho and welcome back. So today we are going to dive into a really fascinating topic, the cholinergic world. It's all about how our body sends messages. We look at this tiny molecule, acetylcholine, and see how it works as a messenger, and more importantly for us as future doctors, how we can use drugs to either make its voice a whisper or a shout. It's a great story of signals, receptors, mimics, and of course, medicines.
All right, class. So here's our plan for today. Simple and logical. First, we'll talk about acetylcholine itself, our main character. Then we'll see where it acts, the receptors. After that, we'll look at drugs that copy it and drugs that make it stronger. And finally, we'll see the practical side, clinical uses and the dangers. Very straightforward. Okay, so let's start at the very beginning, the absolute foundation. To understand any of the drugs, you have to first understand the life story of our main hero, acetylcholine, or ACH as we'll call it from now on.
Now this picture, this is brilliant. It shows the whole story in one go. Just look closely, okay? See, choline comes into the nerve. It combines with acetyl-CoA, and boom, we have our hero, ACH. Then it's packed neatly into these little bags, these vesicles for storage. Simple, right? Now when the nerve gets a signal, these vesicles pop open and release ACH into this gap, the synapse. And from there, it goes and acts on the receptors, you can see them there, labeled N and M on the next cell. But wait, the story doesn't stop here.
So here's the crucial part. This ACH signal, it can't just hang around forever. It has to be switched off, and switched off fast. And that job belongs to a special enzyme, cholinesterase. Just think of it like a super efficient cleanup crew. It comes in and breaks down ACH in microseconds. I mean, it's incredibly fast. And the main type, this acetylcholinesterase, or ACHE, is sitting right there in the synapse, just waiting to do its...
Okay, so ACH is the message, and it's released. But who's listening, right? There has to be someone to receive the message. So let's now meet the two main types of receivers, or as we call them, receptors.
So, the two big families are muscarinic and nicotinic. The best way to understand the difference is with an analogy. Think of the nicotinic receptor like a simple light switch. You flick it, the channel opens instantly, ions rush in, and bang, you get a super fast on signal. Now, the muscarinic receptor, that's more like a fancy dimmer switch in a smart home. When ACH binds, it doesn't just open a gate. No, it starts a whole chain of events inside the cell. A slower, more complicated process. So one is fast and direct, the other is slow, and, well, it modulates things.
And it gets even more interesting, Within this muscarinic family, we have different subtypes, and they do different jobs in different places. For example, the M2 receptors. They are famous for being in the heart. What do? They slow the heart down. That's your vagal bradycardia right there.
Then you have M3 receptors, the real workhorses. You'll find them in smooth muscles, in glands, making things contract and secrete. So you see, in pharmacology, location is everything.
And for the nicotinic receptors, it's a similar story, but simpler. There are really two main places they work. You have the NM type, M for muscle. This is at the neuromuscular junction, where your nerves tell your skeletal muscles to contract. Then you have the NN type, N for nerve. This is where nerves talk to other nerves in the ganglia. So one for muscle, one for nerves. Simple to remember, Hannah?
Okay, so now that we have the basic wiring diagram, let's get to the fun part, the drugs. How can we play with this system? Well, the first strategy is to use drugs that are basically imposters. They look and act so much like ACH that they can fool the receptors. These are our cholinergic agonists. And these imposters, they come in two main flavors. First, you have the choline esters. These are basically chemical cousins of ACH that we make in a lab. And then you have the alkaloids. These are powerful chemicals that come from nature, from plants, from mushrooms, things like that. This table is fantastic. It really shows you pharmacology in action. Just look. At the top you have acetylcholine. See the +++ under hydrolysis? It gets destroyed very, very quickly. Now jump to the bottom to bethanyl. See the minus sign? That means it's not broken down by the enzyme. It resists. And look at its nicotinic action, a big minus. It only acts on muscarinic receptors. So by making these small changes, we've created a drug that lasts longer and is more specific. That's why it's useful for things like the urinary bladder. All right, so that was strategy 1, add an imposter. Now for strategy 2, which is completely different. Instead of adding something new, what if we just stop the cleanup crew? What if we block that cholinesterase enzyme? Well, if we do that, the body's own acetylcholine will hang around for much longer, and its signal will get much, much louder. We're amplifying the natural signal.
Now, these drugs that block the cleanup crew, they come in two types. Some block the enzyme only for a little while. We call that reversible inhibition. These are your carbamate drugs. But then there are others that block it permanently. We call that irreversible inhibition. And these are the really dangerous ones, the organophosphates that you find in pesticides and nerve gases.
So first, let's just appreciate how the system normally works. Just look at this. ACH binds to the enzyme, and it's broken down, hydrolyzed, in a fraction of a millisecond. It's an unbelievably fast and efficient reaction. The signal is sent, and poof, it's gone, instantly cleaned up.
Okay, now look what happens when a reversible inhibitor comes along, like a carbamate. It binds to the enzyme, and the enzyme gets carbamylated. And what does that mean? It means the whole process becomes a very slow reaction. The enzyme is basically stuck. It's not permanently broken, but it will take minutes, maybe hours to recover. We've essentially put the cleanup crew into slow motion.
And now, here's the really dangerous one, the irreversible inhibitor, the organophosphate. This thing doesn't just slow the enzyme down, no. It forms a super strong, stable bond with it. It phosphorylates it. And the enzyme, it doesn't recover. It's aged, it's finished. So the cleanup crew isn't in slow motion anymore. It's permanently out of a job. And that is why these chemicals are so incredibly toxic.
Okay, but Joe, pay attention now. This is a classic exam question. The difference between physostigmine and neostigmine. It's all about one simple thing. Can it get into the brain? Physostigmine, it's lipid-soluble, it has no charge, so it can just walk right across the blood-brain barrier and cause CNS effects. But neostigmine, it's a quaternary compound, it has a charge, it's like it has a big do not enter sign for the brain. So its actions are all outside the brain, in the periphery, especially on skeletal muscle. A very, very important difference to remember.
So, we have these two types of drugs, right? The mimics and the amplifiers. Now let's bring it all together. How do we actually use this knowledge to help patients? And what happens when things go wrong, when the system goes into overdrive? Let's look at the clinical side of things. Now this image shows you exactly what goes wrong in a terrible disease called myasthenia gravis. It's an autoimmune problem. That means the body is attacking itself. You can see these little Y-shaped things, the antibodies. They are attacking and blocking the nicotinic receptors on the muscle. So the nerve releases acetylcholine, but the message has nowhere to go. There are very few receptors left to listen. And the result? Extreme muscle weakness.
So how do we treat this? The logic is beautiful. We use one of our amplifier drugs, an anticholinesterase like neostigmine. What does it do? It stops the cleanup crew. This causes the amount of acetylcholine in the synapse to go way, way up. And this big flood of ACH has a much better chance of finding one of the few healthy receptors that are still working. We are simply amplifying the signal to push through the blockade. It's a simple funda, hai na?
But on the flip side, what happens in a poisoning situation? A farmer gets exposed to organophosphate insecticides. Now you have irreversible blocking of the cleanup crew. This leads to a massive, uncontrolled buildup of acetylcholine everywhere, in the muscles, in the glands, in the brain, everywhere. We call this a cholinergic crisis. It's like a complete storm of overstimulation.
And managing this is an emergency. It has to be done fast. The treatment is very logical, step by step. Step 1, basic life support. Keep the patient breathing, keep their blood pressure up. Step 2, give atropine. Atropine is a blocker. It will block the muscarinic effects, all the secretions and the breathing difficulty. But, and this is the most crucial part, for organophosphate poisoning, you must give step 3, an oxime, like pralidoxime. This amazing drug can actually go and rescue the enzyme. It can break that strong phosphate bond and bring the cleanup crew back to life.
So, Butcho, if you forget everything else from today's lecture, please just remember this one thing. There are two main ways we can play with the cholinergic system. We can either mimic acetylcholine by adding an imposter, or we can amplify its natural signal by stopping the cleanup. Mimic or amplify. That's it. That's the entire story in 2 words.
And that's our session for today. What a journey, huh? From a tiny molecule all the way to life-saving treatments. So as you go, I want to leave you with a thought. We've talked a lot about stopping the cleanup crew, but what if we could go back to the beginning? What if we could find a way to safely boost the production of ACH, or make the nerve release more of it? What would that mean for medicine? Just think about the possibilities. We'll explore more next time. Thank you, everyone.